Good afternoon. Let me welcome you all back to the Wednesday afternoon lecture series, and of course to the NIH. I know the shutdown wreaked havoc with our science here, and we wish this had never happened, but it's good to be back. I know people are happy to be back in the labs, and hopefully happy to be back to hear a terrific lecture today. We did miss 4 of the Wednesday afternoon lectures during the shutdown. Tachi Pa, Shinya Yamanaka, Joe Gall, and Ron Vale. We're not sure about rescheduling Yamanaka because he's mostly located in Japan, but the other 3 talks will be rescheduled. And because we only have a limited number of Wednesdays, they might not be on a Wednesday, so watch your email carefully to find out when those terrific speakers will be talking again. So before we start our lecture today, I'd like to tell you a little bit about George Khoury, in whose memory this lecture series is named. We apologize for not having a program. We normally do for our named lectures, but again, the shutdown wreaked havoc with our plans. Those of you who don't know George, he was the chief of the Laboratory of Molecular Virology at the NCI and died in 1987 at the young age of 43. From complications of lymphoma. Despite this incredibly premature death, he was already a very established scientist working on SV40 as a model system to study gene regulation, how cancer viruses transform cells, and how viruses infect cells. This paradigm-shifting work resulted in his election before his death, even at age 43, to the National Academy of Sciences. George was also known to his friends and colleagues and to his many trainees as a very wonderful mentor and a patient teacher. He taught and showed his colleagues and trainees how enormous energy, combined with a model system which was tractable, could yield incredibly useful information about the biological world. He was enthusiastic about everything and everyone. And I can still see him. He used to sit in the front row. Not many people sit in the front row these days. And he would sit on the edge of his chair. You could see him just about to leap at the speaker to ask questions, which were always insightful and enormously creative. He had impact not only at the NIH, where he trained many people, but also at Princeton, which was his alma mater, where he helped establish the Department of Molecular Biology. Now, one of George's close colleagues during his time at the NIH was T. Zhang, who used to organize this lecture series every year and unfortunately died a little bit less than a year ago. T. himself was a very accomplished scientist and polymath who worked on HTLV1 and unraveled the mechanism by which this virus hijacks cellular mechanisms to enable transformation. He also epitomized many of the characteristics and qualities that George himself had, being an outstanding scientist and a wonderful mentor. He was the Khoury lecturer last year, just a few weeks before his death. We're delighted this year to have Carl Wu as this year's Khoury lecturer. Carl, who spent virtually all of his career at the NCI at NIH, has been for the last year or so a senior fellow and lab head at the Howard Hughes Medical Institute, Janelia Farm Research Campus. Carl was born in Hong Kong, did his undergraduate work at St. Mary's College in California, and received his PhD in biology from Harvard, where he worked with Sally Elgin, following which he was a Harvard junior fellow with Wally Gilbert. And then he came to the NIH, and eventually became chief of the Laboratory of Molecular Cell Biology, and then chief of the Laboratory of Biochemistry and Molecular Biology at the NCI. Dr. Wu is known as one of our foremost contributors to understanding how chromatin structure affects positioning of nucleosomes and gene transcription. He began his work by discovering that DNA's 1 hypersensitivity sites predict sites of transcription initiation in the Drosophila heat shock genes, and also in the rat insulin gene. 20 years ago, he discovered that promoter-specific chromatin remodeling is mediated not only by sequence-specific transcription factors, but also, surprisingly, by an ATP-dependent factor known as NRF, for nucleosome remodeling factor. Using yeast genetics and genomics, he characterized yeast remodeling complexes, and 10 years ago discovered that the yeast SWR1 complex uniquely catalyzes the efficient replacement of conventional histone H2A with histone H2AZ, a variant histone important for transcription. This really was another paradigm shift in understanding transcription regulation, because here was a deviant type of histone which challenged the idea that nucleosomes were based on certain kinds of structures. For this work, uncovering the secrets of how chromatin regulates gene expression, Carl was elected to the National Academy of Sciences, the Institute of Medicine, the American Academy of Arts and Sciences, Academica Sinica, and the European Molecular Biology Organization, 5 of the major honorary scientific societies in the world. His talk today is entitled Chromatin Structure and Gene Expression. Carl.
Well, thank you, Michael. That was very kind. It's my privilege to follow the list of speakers who've spoken at the Khoury lecture, including T. Zheng, whom I knew when I was here. I've been in the audience many times listening, so it's a treat for me to be up here to speak and present our work in George Khoury's honor. I first met George myself in 1981. That was when I was looking for a job, and I came down to Bethesda to visit Maxine Singer's lab, and I remember I knew people in George's lab. Nadia Rosenthal was a graduate student with me at Harvard. So I remember walking into the old Building 41 without having to take a shower, and speaking to George and Peter Gruss, who was a postdoc at the time, and Nadia, and just learning about what they were doing with enhancers, which is what was going on in the 1980s. It was very exciting, and it was part of the excitement about the intramural program that attracted me to come here in the first place. And so, in fact, I showed up in 1982. So George Khoury was one of the discoverers of enhancer elements. And this is a review that George and Peter, by the way, is now the director of the Max Planck Institutes in Germany. They wrote a review about enhancer elements which were defined on the basis of their work, they and 1 or 2 other labs, working on the small DNA tumor viruses. And the importance of that work was to tell us that there was just only a short sequence, roughly 200 to 300 base pairs of DNA, just upstream of the transcription start site, which could enhance transcription when it was put in front of another basal promoter, a basal sequence. And they also showed, they and others showed subsequently, that you could take this and move it around in an orientation-independent manner, as well as putting it at the distance away from the transcription start, and it would still work at a very high level. So, of course, we now know that genomes are full of enhancer elements, hundreds of thousands of them, even up to 1 million in the case of the human genome. But it all started really here in Building 41 with SV40. Now that work not only sort of presaged the great advances that have happened in the study of enhancers, but also the proteins, the protein factors that recognize the sequences within the enhancer elements. And about a few years later, a number of people, and most importantly Bob Tijan's lab at Berkeley, identified the first eukaryotic transcription factors I should point out that this was also going on concurrently with the work going on in Don Brown's lab, who had identified the 5S regulatory element in Xenopus as the first eukaryotic promoter, and the transcription factor TF3A by Bob Rader that recognized the 5S regulatory elements. So anyway, TF3A and SP1 seen here really are the first set of eukaryotic transcription factors. SP1 recognizes the GC-rich element, which is right upstream of the transcription start, and further upstream are the so-called AP series of factors that are also involved in recognizing the SV40 enhancer. These, of course, are host factors. They're not encoded by the virus. They're actually encoded by the mammalian genome itself. And that, in turn, of course, presaged the explosion of studies on transcription factors, sequence-specific factors. And at the last count, I think we now have roughly about 1,600 human transcription factors encoded by the human genome. And they, of course, regulate every gene in the cell, including these master regulators and reprogramming factors, some of which you are very familiar with. For example, MyoD over here is the transcription factor that can switch a fibroblast into the muscle lineage. And the famous 4 here, the Fab4, OCT4, SOX2, KLF4, and MYC, are the 4 Yamanaka factors that can reprogram a fibroblast into a pluripotent stem cell. And of course, there are all these other ones that are coming out of the woodwork now. So this is all very well and good. We've got enhancers. We have transcription factors that recognize the sequences within the enhancers. But there's one small problem, which is that the DNA is not accessible, and many of you know this because this is a hotbed of chromatin research at the NIH. But when the DNA is wrapped in these fundamental particles consisting of 8 histones, as shown in the celebrated crystal structure over here, 4 histone proteins, each a pair, giving you a histone octamer. The DNA is wrapped around the octamer. And when it is so wrapped, a lot of it is not, the information is not accessible. This is Feng Wang in the lab, who's a crystallographer, took some artistic license and put a couple of nucleosomes together as a folded part of a chromatin fiber. And you can see the major groove, and you can put your finger in the groove and read off about 4 or 5 base pairs. But then you run into the histones as you read into the part that's facing the histones. And so a lot of that information is lost. And many factors, in fact, really have a poor recognition of nucleosomal DNA, with a few exceptions. There are some factors that, in fact, do quite well on nucleosomal DNA, and there are even some factors that prefer nucleosomes. But by and large, in general, many of them would much rather have the DNA free. And so what many years ago we found here was really that there was a transient, if not a persistent, rearrangement of chromatin at promoters and enhancers that unravels the DNA and creates this stretch of DNA that shows up either as a DNA's hypersensitive site. This is the one at the heat shock promoter that Michael mentioned, shown by the sort of the old-fashioned southern blot followed by restriction enzyme cleavage. And then these are the cleavages that are induced by DNase 1. It's a fuzzy band, which gives you a sense of this, the stretch, the width, the breadth of the target that's accessible to cleavage by DNase 1. And here again, we've taken some artistic license and put together this 3-dimensional like structure. But this DNA is, in fact, a real promoter. It's the RPL4A gene in yeast. And the fact that it kind of wiggles a little bit like this is just part of the sequence itself. So the question then becomes, well, what creates this open chromatin? And this is really a sort of a universal aspect of chromatin architecture. And we now know, many of you know, that it's the combination of transcription factors, the sequence-specific factors that recognize DNA, that recruit a whole family of co-regulators, these so-called remodeling enzymes, two main classes, those that covalently modify the flexible histone tails, histone modification enzymes, and the second class are the ones that use the energy of ATP hydrolysis to mobilize histones on the DNA and create these nucleosome-free regions. And in fact, if you add up all the proteins that comprise these classes and other related classes of co-regulators, we're coming up to, again, roughly 1,000 or so proteins in the human genome. So we're talking roughly 10 to 15% of genetic information is simply devoted to regulating gene expression on a genome-wide basis. So nowadays, this is sort of a more contemporary view of mapping nucleosomes on DNA. A technique known as MNase-seq, in which one uses an enzyme, micrococcal nuclease, which is a classical enzyme used in the field that chops up DNA between nucleosomes. And instead of doing southern blotting, one would basically sequence everything that is resistant to MNase cleavage and then map it back to the genome itself. And then, because we're always working with populations of cells, this basically shows you what the arrangement would look like for a cell population in one gene. So if, in fact, the nucleosome was extremely well positioned, as this one is, near the transcription start, then every cell would generate a DNA fragment of this size located like so, at this position here. If some of the cells had nucleosomes right next to it, but other cells had it blank, well, then the number of the abundance of resistant fragments would drop, and you would see that in the MNase-seq signal. And if down over here, towards the end of the gene where things seem to be getting scrambled, each cell would give you a different pattern, and if you would then add them all up, you would end up getting, things would start looking very scrambled, okay? You can if you can take all that data, and in this case, we've taken data that came from David Clark and Bruce Howard's labs. It's a paper they published in 2011. This is for the yeast genome, the budding yeast genome. What you can see, we're looking at about 4,500 genes here, and each line represents one gene, and here's the transcription start site, and we've aligned all the genes according to the transcription start. And what you will see as you go down, and these have been arranged by Peter Fitzgerald, our collaborator, in a way such that some genes have zero nucleosome-free region, the DNA's hypersensitive site, and others have very long ones. This one is roughly 300 base pairs. And as you look through the set of genes, you can arrange them in order of increasing lengths of the nucleosome-free region, and this is what you see. And what's quite striking is that in the case of budding yeast, there's this big gap that is basically seen throughout the genome, with the exception of a small group of genes up here, which have no gap. These are the ones that actually are inducible genes, and in fact, it requires signaling to turn the genes on and to create a nucleosome-free region. And you can see a very nice first nucleosome, +1, +2, +3, and as you keep going down towards the end of the gene, things start getting scrambled. Well, it turns out that this kind of architecture is a universal one, not only among eukaryotes, but even in archaea. So here's a summary of all the data sets that came from two labs, Pew Lab and Denislow Lab. And we're looking at now, this is the fission yeast, showing again the alignment is to the transcription start site. And here's the +1 nucleosome. And then right upstream of that is this gap, this valley here. Here's Cerevisiae, okay, so this one looks very nice. But here's the worm, and again, there's a drop here. here's the fruit fly, human, slime moles, Arabidopsis, which is a little pepper plant, and even archaea. This is an organism that lives in hot springs, and they have archaeal histones, histone-like proteins. Even their start sites, right upstream, have a region which is free of the archaeal histones. So this seems to be really a universal architecture of chromatin in many, many organisms. So there's one additional aspect of this universal architecture, which is really the subject of my talk today, and that is a histone variant called H2A.Z. And that's also found at the so-called +1 nucleosome, along with a lot of modifications such as acetylation and methylation of the histone tails. So what is histone H2A.Z? It is a variant histone that I'll tell you a little bit more about, but it is located, as I mentioned here, at the +1 nucleosome, a little bit at the -1. The structure of the H2AZ nucleosome, which I think you can barely see in the light, is very much like the canonical one, and there are just some very subtle differences in the overall shape. However, as you will see, that there are certain amino acids which matter a lot, and those are the ones which make a difference, make a functional difference in conferring novel properties to this nucleosome. If you were to look at the distribution of H2AZ by MNA-seq, as Frank Pugh did, this is his data set, this is what you see, that there is in fact an enrichment of the H2AZ at the +1 nucleosome downstream of the nucleosome-free region. H2AZ was discovered at the NCI. It was discovered by Michael West and Bill Bonner. Bill Bonner is still here. I don't know if he's in the audience, but he's in building 37. And what Bill discovered was really a whole family of histone H2A-like proteins. And he discovered this by a series of manipulations of gel conditions, which in those days was the only way one could use to distinguish proteins that were relatively closely related. And what he did was he purified what we all thought was a homogeneous population of histones and ran them on these triton acid urea gels. And he discovered that there were a whole number of variations on these proteins that showed up as discrete bands. And the second variant he called H2A.Z, H2AX, which is related to DNA repair, is the other one that he discovered. H2AZ comprises about 4% of the total H2A in mouse L cells. A fraction of each of these histones is combined with ubiquitin. And the molar sum of these 8 species approximates the moles of H4, H2B, and H3, which are the other histones. I should just point out there's yet another histone variant that Bill did not discover, which is called SEMP-A. It'll come back at the end of the talk. But that's a histone H3 variant. So the main histone variants are really the H2AZ relative to transcription, the H2AX for DNA repair, and the SEMP-A histone H3, which is related to the centromeric histone. Anyway, this was published in 1980 in Biochemistry, and I confess it wasn't, I would say, something that we all paid attention to. But that dramatically changed. It changed because in the 90s and in mid and late 90s, knockout technologies were applied to the histones. And because H2AZ was a unique gene, one could knock it out. And that's when things completely changed. It turns out that this is a very important histone. It's functionally required. It is conserved from yeast to human. And if you knock it out, in a metazoan, either a fly or worm or a mammalian organism like mouse, you basically don't survive, and the time of death is generally early in development. And then molecular studies by Mitch Smith's lab especially discovered that this was in fact the histone H2AZ that was found at the promoters. And so this very minor fraction, this 4% or so, turns out to be the most important 4%, because that's where the action is at gene promoters and enhancers. And as Gary Felsenfeld showed here at the NIH, it's also at the insulators. And in fact, it's at every element where every functional element in the genome in every organism. And that includes origins of replication as well. So this is really quite a signature. And so what does it do? So there's been quite a bit of work done in biophysics as well as biochemistry. It affects the way the chromatin fiber folds. It affects the accessibility of the factors in the vicinity of the H2AZ nucleosome. It affects nucleosome dynamics. That nucleosome, that +1 nucleosome, is particularly quote-unquote hot in the sense that it just gets exchanged a lot, much more than the rest of the nucleosomes along the gene. And there's other biochemistry that shows that it affects the kinetics of transcription and even non-coding transcription as well, as was founded by Shiv Grewal's lab here at the NCI. So this turned out to be a very important histone. Well, how did we get into this? Well, by accident. So the question becomes, how is this histone put into the +1 nucleosome? And as Michael mentioned to you, we'd been studying this whole family of ATP remodeling enzymes, and we were systematically purifying them and characterizing their components. And we had switched from fruit flies to yeast because it was just more convenient to work with yeast, which was both genetically and biochemically accessible. So in purifying one of these enzymes, called the SWR1 enzyme complex, I refer to this as SWR1, you get a whole bunch of bands on the gel, which is fine. And in those days, this was before molecular proteomics really took off. One had to cut out the bands from the gel and send them off to the mass spec, which is what we did. And what Snow Shen, who was one of the postdocs working on the project, did for completeness was he cut off the bottom of the gel. And usually the bottom of the gel is stuff that you throw away. Unfortunately, he didn't do that. The bottom of the gel is here. You can barely see the band. But in fact, when we sequenced the bottom of the gel, not only do you find histones, because they're small proteins and they're all running towards the gel front, what he found was that there was this H2AZ that Bill Bonner had discovered long ago, which was highly represented in that population. And it was only the SWR1 purification amongst the 8 or 9 enzymes that we purified that had that enrichment of the H2AZ. So that suggested to us that SWR1 had to be in some way connected to the biochemistry of H2AZ. And the most sort of the simplest explanation would be that in fact it could have been important for depositing H2AZ at that +1 nucleosome. And so Gaku Mizuguchi, who was the lead author of this paper in 2004, in fact worked out an assay to show that. And this is just to show amongst all the members of that remodeling enzyme family, all of which contain this so-called SF2 superfamily of DNA-dependent DNA translocases, which are ATP dependent. This is the one that is involved in H2AZ metabolism, and it is found from yeast to human and flies and worms as well. Anyway, so let me give you a little bit about H2AZ. So this is how we saw it in 2004. And then Wei-Hua Wu in the lab as a postdoc spent a couple of years working out how this enzyme is assembled by a combination of genetics and biochemistry. It's basically figuring out what were the true subunits, what were ancillary subunits, and how did they assemble, which were the ones which were most important, which ones were less important. And this is simply a summary, and don't worry about any of these names. Here's that ATPase itself, the big protein up here. And it serves not only as the catalytic ATPase, but it's the scaffold on which all the other subunits land, okay? Now, from 2009, and in fact even before that, we were engaged in a collaboration to solve the structure of this enzyme. It's 14 subunits. one megadalton in size. And finally, in 2013, this year, our collaborators at Harvard, Andres Leshner and his graduate student, Vu Nguyen, presented this structure to us, which is another view of the enzyme. And I'll come back to this a little bit more, but basically, it gave us at least some idea of what it looked like physically and allowed us to now begin to hang some of these modules onto the structure itself. I have to point out that this is simply the first view. I cannot even tell you what the resolution is because it doesn't make that, it's not that meaningful at this stage of the analysis, but at least it's something for us to look at. And the structure tells us that there are, in fact, certain modules that mirror the genetic modules that we saw before. So, for example, this region here contains these so-called RVB proteins, and there are 6 of them forming a ring-like structure. And then this region here, the blue one, is in fact the catalytic ATPase. That's SWR1 itself. This module here contains 1, 2, 3, 4, 5, 6 proteins shown here. We cannot even distinguish the individual subunits yet. And this module contains these 4 proteins. What is interesting, and I have no explanation for this now, is that there are 1, 2, 3, 4, 5, 6 subunits that bind to ATP. This is an ATP hydrolyzing enzyme. It needs it to work. But why do we need 6 subunits that bind ATP is unclear at the moment. The catalytic subunit, of course, binds to ATP. The basic reaction that Gaku worked out is shown here. We take the enzyme, we take the H2AZ, which comes as an H2AZ H2B dimer. It comes along with its own chaperone called chaperone for H2AZ or cheese 1. And this has been solved by Yawen Bai's lab at NCI. And we also give it a canonical nucleosome containing 2 H2A H2B dimers, and it's attached to a dynabead. You mix these together and you add ATP, and what happens is that the A gets kicked out and the Z gets put into the nucleosome. And this is shown in a reaction here. The Z actually has a flag tag on it, so we can follow the H2AZ in yeast. It's called HTZ1. And you can follow this on a Western blot. After you've done the reaction, the nucleosome arrays can be pulled down on the dynabead. And now you see an accumulation of this flag on the bead. Only when you have the enzyme and you have ATP present and you have the H2AZ H2B cheese 1 trimer. All of the above gives you the signal, on the replacement signal. And this is a depletion from the solution here. If you add any of these inhibitors of ATP, the reaction is killed. This is a background signal. And most importantly, if you make a mutation in the ATP binding pocket of the SWR1 catalytic subunit, and you purify that mutant enzyme, and enzyme assembly is fine with the mutation in the ATP binding pocket, but the enzyme is dead. So this established sort of the basic reaction that you could recapitulate in vitro under relatively physiological conditions. And of course, in vivo, we and others showed as well that if you knock out any one of these components, the enzyme does not work in cells, and H2AZ is not deposited at that +1 location. So over the last couple of years, we've now been going to the second round of analysis of this reaction. And this has been work done by Ed Look, former postdoc who is now at Stony Brook, and Anand Ranjan, who is still a postdoc in the lab here. And what we see now is we've sort of turned this into a cycle where you have the enzyme. The enzyme has 3 substrates. Okay, it has the nucleosome as a substrate, and it has to bind to that substrate. It has the H2AZ as a substrate. It has to bind to the H2AZ substrate. In fact, there are 2 subunits that bind to H2AZ, 2 locations. And the third substrate is ATP itself. Okay, and only when you have all 3 substrates do you have then an activation of the ATPase, which you can see here. And with this activation, what happens is the loss of one H2A and the insertion of one H2AZ. And this is a second generation assay that Anand in the lab has developed, in which you start off, and we're looking at a mononucleosome now, with just the conventional two H2As. And as you go through the reaction, and this takes roughly 30 minutes to reach completion, you get one step, you get one H2AZ, and the reason we see an upshift in this native acrylamide gel is that the Z flag confers a mobility shift on the nucleosome. And when you have two flags, you shift again. And so you can see it going first to the one intermediate and then to this double substitution intermediate here, the final product here. And so the way we think about this is that you go through one step, this intermediate dissociates, stochastically we would imagine. It rebinds, but now it rebinds to the A phase, because only upon recognition of the A phase does the ATPase get activated. If it binds to the Z phase, it does not. So this actually is a mechanism for terminating the reaction when you end up with a double Z product. So there are lots of things that we need to know about this reaction, and I'll tell you more about this towards the end of the talk. But this is basically the way we can see how the system is working. So the next question is, so now we have the enzyme, we have the substrates, we know how it works in vivo, we know how it works in vitro, but how does the enzyme know how to find its target? And the target is every promoter in the yeast genome. Yeast do not have enhancers, so all the nucleosome-free regions are either promoters or origins of replication. And so you can imagine that there are three mechanisms that could be involved. First, it could be that there are sequence-specific factors that recruit the enzyme, like these factors recruit all the other remodeling enzymes. And for example, there's a protein called Reb1, which has been implicated. But we felt that this was not a good way to design the system, because then you would have to have lots of transcription factors all carrying some domain that attracts the same enzyme. Another possibility is that the enzyme is simply tracking the universal state of the promoter architecture, which is that there are histone modifications occurring at promoters in general, such as histone acetylation. And in fact, there's genetic evidence that those acetylation sites do matter to some extent for the recruitment of SWR1. But the last possibility is that it's simply the nucleosome-free region itself, which is the architecture that is being recognized by the SWR1 enzyme. And it turns out that, in fact, this turns out to be the most dominant mechanism. This is there, but not quite so dominant. And there's absolutely no evidence in an unbiased screen by other labs which have looked for this enzyme associating with any transcription factor in the yeast proteome, and nothing shows up. So let me just then take you through a series of these experiments, which were conducted by Anand Rajan in the lab. And his work was just published last month. And Anand is also looking for a job very soon. So what Anand decided to do was to test this directly by reconstituting a promoter structure in chromatin as a snapshot. That is to say, we're making a gap flanked by two nucleosomes. This doesn't mean that this is always going to be the case, but from a transient point of view, as a snapshot, this is likely to exist. And we can then freeze this snapshot by making an artificial construct. So the way to do this is to take the natural gene promoter, which has anti-nucleosomal sequences, and then two sequences which are extremely high positioning for nucleosomes, the so-called 601 sequence by John Whittam. With this two nucleosomes flanking the RPL4A promoter, one can then go through the standard salt dialysis procedure with recombinant bacterial histones and this DNA, and then generate a series of reconstitutes. And what you see is free DNA, DNA with one nucleosome, DNA with two but not well spaced, and then DNA here, which is the product that we want, and above that, there's DNA with 3 nucleosomes because there's enough room to fit a nucleosome even in the nucleosome-free region. And the trick is to put lambda DNA together with this mixture so that the lambda competes away for all the stuff that you really don't want. And under these conditions, only the species that contains the dinucleosome with the long linker survives. You can purify this on a sucrose gradient, and you can image this. This was sent up to Chris Woodcock at UMass Amherst, and you can see these dumbbells consisting of these dinucleosomes here. You can now take this preparation and label that with 2 different colors. We call this a long linker with the RPL4A promoter in red, Psi5, and then a control dinucleosome that contains just a small piece of random DNA inserted between 2 nucleosomes in green. And then in the same mixture, you can ask, how does the enzyme bind? Does it prefer one over the other? And it's quite clear that the enzyme much prefers the red long linker dinucleosome over the green short linker dinucleosome, and these are the binding curves. And this binding then, in fact, has a functional consequence because you can see that the long linker dinucleosome in this, now this is a functional assay looking for the 1Z or the 2Z products, showing that when you have long linker, the reaction goes much more efficiently than with the short linker dinucleosome, as you can, and here are the binding curves again. All right, and now you can take a look at the image, the enzyme on the promoter. So here we're deliberately taking a long linker dinucleosome, which has been undersaturated for enzyme. So we have a lot of free, unbound dinucleosomes and only a little bit of the bound. And when you image this on the EM, what you can see in the field is that you've got some of these dumbbells like you saw before. These are the unbound dinucleosomes. And now here you see a big blob here. Turns out to be the enzyme itself. And you can measure the length of the linker, and it turns out that, in fact, when you do this for the whole population, there's a very clear invasion of the linker by enzyme binding, either by occlusion or actually direct recognition, that gives you a much shorter free DNA here versus here when the enzyme is not bound. And then if you send this up to the STEM microscope in Brookhaven, you can measure the mass of these particles, and that measurement comes to 1.2 milliadaltons. 1.0 is the calculated mass of the enzyme itself with the 14 subunits, and the 0.2 comes from the nucleosome, 200 kD nucleosome complex. And so this is actually very consistent then with the ratio of 1 enzyme to 1 nucleosome in this complex over there. All right? So now, biochemically, you can see this even better also. You can give the enzyme 3 different substrates, free DNA, a particle, what we call the nucleosome core particle that has no linker DNA at all, and now a core particle with 40 base pairs of linker. And the enzyme prefers, in fact, this substrate much better than the particle or the DNA alone. And here are the binding curves. So you really don't need a dinucleosome. You just need 1 nucleosome with a long linker. And then, in fact, you can say, well, you can go through the whole series of linkers. This is, again, Anand's work. And there seems to be a big jump once you go up to 60 over here in preference for the substrate. So 60 seems to be a magic number that the enzyme prefers. And if you now just give it, forget about histones, just give it the DNA, and we have a ladder. Now this is your commercial DNA ladder. You ask, well, this is a DNA binding protein. So you ask, well, is it binding, and does it prefer long pieces? And of course it does, like any good DNA binding protein should. But when you measure this, you see what the preferences are. The preferences are that it much prefers things that are at least 50 and 60 compared to 40, 30, and 20. So again, there's this jump. So it's as if the enzyme can read the length of the nucleosome-free region and preferentially interact with that structure. Now, that's just for recognition, for recruitment. Once you've recruited the enzyme, do you really need the long linker? You don't. So here's an experiment that Anand did, where he just gave the enzyme the core particle by itself with no DNA sticking out. And in this second generation assay with the 1 flag, 2 flag, you can see Z incorporating onto that nucleosome quite well without any linker DNA at all. So the linker is only for recruitment, not for activation, post-recruitment. So coming back to, so when the enzyme likes to, when it's for recruitment, it likes to have a stretch of DNA flanked by 2 nucleosomes, or at least 1. Does this correlate to anything in vivo? So we went into the literature and compared, and this is what I showed you before, the data set from David Clark and Bruce Howard's labs showing the yeast, the distribution of yeast nucleosomes in the nucleosome-free region here. And now the same order of genes are used to arrange the data set from Frank Pugh's lab, and we're now looking at chromatin immunoprecipitation of the SWR1 enzyme on that promoter, on these promoters here, in order of nucleosome-free region length. And what you can see is that this is a reasonably good correlation that as you increase the length of the NFR, you increase the abundance of the enzyme up to about 200 base pairs, and then it actually drops. This region here, you can see as this drop here, it turns out that the way we normally map nucleosomes is a bit overstringent. And there are nucleosomes that are actually fragile, so-called, which can be eliminated by exhaustive MNase digestion. And if you don't exhaustively digest with MNase, as these workers did in this paper published in 2011, you get the same kind of architecture, but where you now have a big gap here that's longer than 200 base pairs, there's in fact a fragile nucleosome inserting in here. So that, I think, explains why we think that there's a drop here. In fact, that the nucleosome-free region is no longer than about 200 base pairs. Anything longer than that, there are actually nucleosomes there, but they're fragile. So this is interesting. This is just one enzyme. We looked at a bunch of other enzymes in this sort of correlative analysis. And for example, another remodeling enzyme called RISC has no correlation with the nucleosome-free region in terms of its distribution, its abundance in relation to the length of the NFR. But interestingly, TF2B, which is a component of the preniciation complex, has a very good correlation. And TBP, which is another member of the preniciation complex, also has a very good correlation to NFR size, which implies at least the possibility that, in fact, a lot of other enzymes out there use the length of the nucleosome-free region as one mechanism for recognizing where it should be recruited, to where it should be recruited. Okay, a little bit more biochemistry. So who's responsible? So there are 14 subunits. Which of these are really important to recognize that long linker next to the nucleosome? And it turns out that, again, don't worry about any of these names, that there's just one subunit in pink here, which is the most important one. And we could perform this analysis because, again, from Wei-Hua Wu's earlier work, we knew you could cut the enzyme in half into an N-terminal half and a C-terminal half that also mimics that cryo-EM structure I showed you. And the enzyme would not fall apart. And these subunits would go along with the N part, and these subunits would come with the C part. So, and Debbie Wei and Anand purified these mutant enzymes. And now you can ask, who's responsible for recognizing the nucleosome-free region? Here's that long linker dinucleosome. And here's the wild-type enzyme. Here's the N part. It fails to bind. And here's the C part. It does a pretty good job for binding. Of course, the mobility has changed because you've got half the enzyme. And if you knock out, in this complex, if you take the wild-type enzyme and just knock out this pink subunit, you fail to bind. So at least that told us that this was a very important component of recognition. And if you make that knockout, you also can show that the binding of the enzyme in cells is impaired. This is the chromatin immunoprecipitation experiment showing SWR1 enrichment over 4 or 5 genes in the genome. And this is a wild type signal, and then these are the mutant signals for the SWIG2 subunit. So in vivo, SWIG2 is also required for recognition by SWR1. And if you go through 3 chromosomes, average them all out again. This is the wild type signal for SWR1 for the genes on those 3 chromosomes, and this is the green line is the mutant signal. Okay, so now we're coming to the next mechanism. So up to now, it seems like DNA binding, that nucleosome-free region is the most important, is important. But is it more important or less important than histone acetylation? And it turns out in this comparison that acetylation is less important compared to the DNA binding. But so how do we do that experiment? So now we go back to the dinucleosome, and now we acetylate the dinucleosome. Others have shown genetically that the enzyme NuA4 is important for this reaction. And then purified the enzyme with Debbie and acetylated these nucleosomes here. And then in red is the control, which has not been acetylated. And now in the same reaction, you ask, does the enzyme prefer one over the other? And you can see this is what you had been seeing all along up to now. Nothing has been acetylated. But now if you add on acetylation, you can improve the binding, as you can see here in this binding curve. So acetylation is important, and it does help. But now you ask, who's more important? You take that enzyme that we can cut in half. And this half of the enzyme contains the DNA binding component, as I showed you. And this half of the enzyme contains this protein called BDF1, which has a number of bromodomains that I'm sure many of you are familiar with that recognize acetylated histone tails. And you can take this piece and this piece, both at 15 nanomolar concentration. And here's the acetylated dinucleosome. And you can ask who does the better job. And it's clear that it's the C-SWR1 that gives you the stable complex that contains the DNA binding component. The N-SWR1 can show some weak binding, giving you the smear, but nothing quite like the stable complex that you see here. So the bottom line is that the DNA binding dominates over the acetylhistone binding. And we think this is relevant, generally speaking, because there's been a lot of discussion in the community about whether gene regulation is basically dominated by histone modification signals, or whether it is dominated by the DNA sequence that is read by the sequence-specific transcription factors. And I think this is probably, if I'm not mistaken, the first time that one can actually measure the relative preferences and the relative importances of one over the other directly in vitro without any interfering components. And basically, that there's cooperation between the DNA binding and the acetylhistone binding components, but it's a hierarchical cooperation in which the DNA binding dominates over the acetylhistone binding. That's really one of the sort of general lessons we've taken away from this work. The second is that, yes, we've been, for many years, we've been talking about nucleosome-free regions in DNase hypersensitive sites as providing accessibility to chromatin factors. The take-home lesson to me is that, yes, that's true, but it's a little more subtle than that. That is, not only is it simply open and shut, but it's the degree of being open that also seems to play a role. So the degree of chromatin accessibility from either just altering the structure of the nucleosome without moving it around, and there are examples of this, as you know, all the way to creating a free region that is completely devoid of nucleosomes. This provides a way to calibrate recruitment of chromatin enzymes. And I think this may well, be a theme that's going to appear for other chromatin-based reactions. So in the case of SWR1, we think that there are two most important components. The DNA binding component SWIG2. There's a human version of this called YL1. And that's the one that can sense this 60 base pair or more region next to one nucleosome. And then there's this other subunit, BDF1, that binds to the acetylhistone tails. And together, I think they're responsible for bringing this enzyme to the +1 location in the case of yeast gene promoters. There are human versions of this, of course, and I would imagine that such principles would also apply, with the addition that in the case of mammalian systems, transcription factors do also recruit the human version of the SWR1 complex. So the two mechanisms are not mutually exclusive. So let me just quickly then recapitulate where we are now. We have a crude structure for this. Of course, we have a good structure of that. And as I mentioned, Yao and Bai's lab has solved the Cheese1H2A's H2B structure. There's a crude structure of the enzyme bound to the one substrate, the nucleosome. The next challenge is to get the structure of the enzyme bound to both substrates and bound to ATP analogs and see what the structure of that might look like, because we really want to know how the enzyme manages to pull apart the nucleosome, to pull out the H2A and put in the H2AZ. A lot of this is going to involve a lot more hardcore biochemistry, structural biology, and also single molecule methods, single molecule biochemistry to be able to get a hold of the kinetics of these reactions. One of the approaches that we're beginning to work on now is to begin to look at the dynamics of the H2AZ histone in live cells and to use a variety of microscopic techniques that my colleagues at Genelia Farm have been developing to begin to address issues about how long does it take to get an H2AZ into a nucleosome, how long does it take to get an H2A out of a nucleosome. And what you're looking at here is simply single molecules of H2AZ that have been fused to a photoactivatable protein called dendra. And we're imaging all the little blinks that you're seeing are basically single molecule images of H2AZ dendra that are showing up. And this particular microscope, it's called a multifocal microscope operating under the PALM mode in 3 dimensions. What it can do is it can take 9 Z sections in one exposure. So you don't have to go through 1, 2, and then go back to generate the series. It's all done in one shot. And that's particularly useful if you want to capture dynamics of individual particles. I have to say that this was done by Jan Wisniewski and Gigi Chen at Genelia. This hasn't even made it to our lab meeting. So it is basically raw data and completely unfiltered. But I just wanted to show this to you to give you the flavor of what we would like to do, which is to try to image the individual molecules of H2AZ dynamics in vivo. Now, what one does is to take, these are in live cells, and one takes many more frames than just 2,000, or 250 in this case. And then that gets, there's a massive computational involvement to generate basically locations of these particles. And since the shutdown, we also got shut down, at least my side of the lab here got shut down. So we don't have all of that for you today, but we do have the other histone, which is a centromeric histone, CSE4, or SEMP-A. And what you're looking at, again, in budding yeast are single molecules of SEMP-A, which are arranged, located at the centromeric nucleosomes of yeast. Yeast has 16 chromosomes. Each chromosome has one nucleosome that contains a SEMP-A histone. And you're looking at individual SEMP-As in the yeast nuclear volume, which is roughly the size of this box here. And what we find really amazing about the technology is that to within 20 nanometers, which is two nucleosomes width, you can localize the positions of the individual histones with this microscope. So again, this is static. This is not dynamic. but it's simply the foundation of where I think we would like to take both projects with both CSC4-SEMP-A and H2AZ. So let me then just try to wrap up. Gene regulation in eukaryotes, from the textbook models that we studied when I was in graduate school with the Jacob Monod, the famous Jacob Monod papers, of activators and repressors binding to certain targets. Yes, that does operate also in eukaryotes. And there's a lot more of those factors, as I mentioned, in mammalian genomes. But there's a whole layer that's put on top of that, which matters a lot and which plays a intrinsically important role in regulating genes. The enzymes that use ATP to mobilize histones, the enzymes that put in chemical signals onto the histone tails, the variants of the histones that Bill Bonner discovered here, the chaperones of the histones. I didn't even have time to get into the chaperones with the exception of the cheese 1 that I mentioned briefly. The whole realm of non-histone architectural proteins, such as cohesins, condensins, HMG proteins, also are important for eukaryotic gene regulation. Signals on the DNA methylation, I'm sure you all know, demethylation, the enzymes that are involved in those processes play an important role for many metazoans, but not all. They're not found in yeast or Drosophila, but still very important. And then lastly, the whole RNAi machinery that for many organisms is brought down to the chromatin to help in regulating its activities. So it's a lot more complicated, but I think mammalian metazoans are more complex than bacteria, so not too surprising. But it does say that when we look back and think about transcription factors and the ones that are now very fashionable, that in order for them to work properly, it's very important to understand the chromatin context under which they operate. And again, all those principles that laid out in the previous slide show up again here in a very recent review by Hochlidinger. So let me then thank the people who've been contributing to our own work. For the past 10 years, we've been working on H2AZ, and we've had some really great postdocs and research associates in the lab. Many of them have moved on to other labs, moved on to their own labs in some cases. The current crew, Anand Ranjan, I mentioned to you, has been a postdoc for a number of years in the lab and hopefully will soon be a faculty somewhere. Anand's work basically was a collaboration between him, Debbie Wei, who's our research assistant, Feng Wang, who recently joined the lab, a crystallographer, and Gaku Mizuguchi, who moved with me to Genelia. These are the people who, he started the H2AZ project along with Snow Shen, and he's doing other things at the moment. Jan Wisniewski, I showed a little bit of his work at Genelia about imaging the single molecules. Has been working also with Hua Xiao, who's been doing some hardcore biochemistry on the centromeric nucleosome, which is a story in and of itself. And maybe some other day I will share that with you. Our collaborators, Peter Fitzgerald, NCI Genome Analysis Unit, all the bioinformatics with Peter. Electron microscopy, Chris Woodcock, who was one of the two labs that, two people who discovered nucleosomes in the 1970s. We got Chris out of retirement to image our nucleosomes. He's still at UMass Amherst as emeritus professor. Ed, Snow, Joe, Weiwa, former postdocs now running their own labs. Our collaborators, Vu Nguyen and Andres Leshner at Harvard, who did the cryo-EM structure for SWR1, Yawen Bai Zheng Zhou at NCI, who did the NMR structures for the histone chaperones, Maxime Dahan, Bassam Haj, and Gigi Chen, who are people at Genelia. Maxime and Bassam have returned to Paris, who've been working on the MFM, and we've been collaborating with them, as you saw. So thank you very much.
So I'm sure Carl will be happy to answer some questions. Yes?
Carl, I have two questions. So the first one is that it appears the yeast H2Z is the function is slightly different from mammalian ones, because the mammalian H2Z is required to maintain the chromatin accessibility, particularly enhancers. But in yeast promoters, it appears it explores or exploits the nucleosome free regions. So that means the nucleosome accessibility or the chromatin accessibility does not require H2Z there. How do you explain this? And then what maintains the nucleosome free region?
Yeah, I mean, that's a good question. I think I don't think the yeast people have looked at the accessibility in exactly the same way as, for example, you had in the mammalian system. And so it's probably worthwhile to redo those experiments, because there may be a component of accessibility that also plays a role in the yeast case. But aside from that, the point I think that's, and I thank you for the question, which is that the yeast enzyme SWR1 takes advantage of a pre-existing nucleosome-free region. And so who makes that nucleosome-free region is probably other transcription factors and other remodelers. And in the case of the ones that I showed you, we know that, for example, REB1 and RISC actually play an important role in creating the nucleosome-free region first. And then SWR1 comes in later to take advantage of the nucleosome-free region. And I suspect that sort of sequence of events is probably going to be general.
So in the genes, there is nucleosome-free region near the promoter, and sometimes I have read that the first splice junction also shows the NFR region, which is a smaller. So how much different that region compared to this promoter-based NFR region?
Can you repeat that, because I didn't catch you?
So the first splice junction amongst the gene also shows the nucleosome-free region, NFR.
The splice junction?
Yeah, first splice junction.
I'm not sure I follow that.
Okay.
We could talk later. Maybe that would be the best thing to do. Hi, Wing.
Hi, it's a wonderful talk and a beautiful work. Just one question. So you showed very clearly that SWR1 recognizes free DNA and the acetylation helps in binding. What about the H2A, H2Z exchange? How the acetylation effect, does it have more effect?
Yes, so it also, I didn't show the data, but in fact it does
Should have, because it's a
yeah, I mean, simply because it helps the recruitment, and therefore, what we have not done, and maybe Anna can correct me if this, we have not done the experiment, but you bypass recruitment completely.
Yeah, recruitment is secondary. I was thinking
and
about
then
the
just see how acetylation affects the second part of the reaction. That may have been done, maybe not. If not, it should be done.
Right, because it should be the effective specificity. The substrate is right or substrate is not. So it will be beyond the effect of the binding. It should be more on the ATPase stimulation and on the subunit exchange.
And exchange, yeah. No, that's a good question.
Okay, we will have a reception in the library sponsored by the FAS. You can ask Carl more questions. I also want to point out that next week, Wednesday, Thursday, and Friday, we have rescheduled the Research Festival, so I hope you can all attend those events. And that will include the next week's Wednesday afternoon lecture, which is being given by Erica Sapphire on the structural basis of Ebola viral pathogenesis, which will be next Wednesday at 3, embedded into the Research Festival. So the end of next week, expect to have a lot of great science at the NIH. Thank you all, and we'll see you at the reception. Also next week, Randy Sheckman, this year's winner of the Nobel Prize in Medicine or Physiology, will be giving a talk on November 4th at what time? 11 a.m. here.